Dear students, today we study about seed dormancy. Myself, Dr. Santosh Madale of Sri Yashwantrao Patil Science College, Solapur. The seed dormancy topic is divided into introduction, types of dormancy, types of seed dormancy, causes of seed dormancy, overcoming of seed dormancy. First of all, we try to know about what is dormancy. Dormancy is nothing but during the developmental cycle of the plant, at some phase, certain structures like buds, tubers, seeds go through a period of temporary suspension of growth activity or slows down for a period of time or deep sleep. Such a state is called dormancy. So this minimizes the metabolic activity and therefore helps an organism to conserve to energy. So dormant is nothing but not active or growing but able to become active later. Waring defined in 1969 dormancy as any phase in the life cycle of a plant in which active growth is temporarily suspended. Seed dormancy is nothing but it is the incapacity of fully developed mature viable seed to germinate even under favorable conditions. In such cases, the completely dry ripe seed is physiologically inactive and is said to be in a resting phase. So the seed is called dormant and the phenomenon is termed as dormancy. The main reason behind this condition is that they require a period of rest before being capable of germination. This condition may vary from days to months and even years. Then types of dormancy. According to Waring 1969, the dormancy may be divided into mainly two types: imposed dormancy or quiescence and innate dormancy or rest. The imposed dormancy means the dormancy due to unfavorable environmental conditions is called imposed dormancy or quiescence. Innate dormancy means the dormancy due to conditions within the dormant plant or organ is called innate dormancy or rest. It is a condition in which the germination or growth fails to occur even though the external environmental conditions are favorable. During the entire process, there may be a following three phases of dormancy: pre-dormancy or early rest. During this phase, the dormant organ has capacity to resume growth by various treatments. This capacity of germination or growth is not completely lost. It is called pre-dormancy. Full dormancy or mid-rest, when a seed or organ become completely dormant and germination or growth cannot be induced by changes environmental conditions, it is called full dormancy or mid-rest. Post dormancy or after rest, when a dormant seed or organ gradually emerges from full dormancy and in it the germination or growth can be induced by changing environmental conditions, it is called post dormancy or after rest. The dormancy may be true, relative or secondary. True dormancy means when a seed or organ, the germination or growth cannot be induced under any set of environmental conditions. It is called true dormancy. Relative dormancy means when in a seed the germination can be induced under the specific conditions even at the time of its deepest dormancy. It is called relative dormancy. Secondary dormancy means when in a seed has not fully emerged from dormancy and is again thrown back into the full dormancy by certain environmental conditions, example temperature, it is called secondary dormancy. Then types of seed dormancy. Crocker 1916 divided seed dormancy into two types: seed coat induced dormancy and embryo induced dormancy. The dormancy of seeds due to extreme hardness of seed coat is called seed coat induced dormancy. And embryo induced dormancy means the dormancy of seeds due to rudimentary or complete dormant embryo is called embryo induced dormancy. According to Baskin and Baskin, 1998 and 2004 have proposed a comprehensive classification system which include 5 classes of seed dormancy: physiological dormancy, morphological dormancy, morphophysiological dormancy, physical dormancy, and combinational dormancy. Physiological dormancy means physiological dormancy prevents embryo growth and seed germination until chemical changes occur. It is a result of seed requiring some type of physiological conditions to be met in order to germination. So these chemicals includes inhibitors that often retard embryo growth to the point where it is not strong enough to break through the seed coat or other tissues. Morphological dormancy. This class of seed dormancy refers to seed with underdevelopment and differentiated embryos. This includes embryos in which the cotyledons and hypocotyls radicals are differentiated but small in size. These embryos do not have physiological dormancy and only require additional time to grow and germinate. Commonly under favorable conditions, embryos in such seeds begin growth within a period of from a few days to several weeks, and seeds germinate within 30 days. Morphophysiological dormancy. In this class, the seeds have embryos that are underdeveloped in size but differentiated into cotyledons and hypocotyls radical as well as physiological component to their dormants. So thus to germinate these seeds require time for growth, embryo growth and a dormancy breaking treatment. Morphological dormancy can be divided into 2 types: epicotyle dormancy and double dormancy. Physical dormancy means the seed coats are impermeable in water due to macroscleride cells, mucilaginous outer cell layer, or hardened endocarp. Depth of the puncture to the seed coat increase, so did the permeability of the seed coat of water. Example: olive, peach, plum, cherry, etc. Combinational dormancy. This class groups seeds with simultaneous physiological and physical dormancy. In this case, physiological dormancy is generally characterized as non-thief and a cold stratification treatment of seed after scarification to permit imbibitions is a common dormancy breaking treatment in this class of seeds. Then next point is causes of the seed dormancy. Bewley and Black 1994 have divided seed dormancy into 2 categories: seed coat based and embryo based. The dormancy of seeds may be either due to single or combinational of many different following factors. Seed coat induced dormancy or hard seed coat. The seed coat is mostly formed by the integumentary layers of ovules. The seed coat consists of 2 layers. The outer is called testa and the inner is called as a tegment. The testa particularly is composed of a complex mixture of polysaccharides, hemicellulose, fats, waxes, proteins. During the maturity of seed dehydration takes place in the seed coat and thus it forms a tough protective layer. The hard seed coat prevents germination due to following reasons. One is water impermeability. The seed coats of many plants belonging to the family Leguminaceae, Chenopodiaceae, Malvaceae, Convolvulaceae, Solanaceae, and Nymphaceae, etc. have very hard seed coats, so it cannot permeable to entry of water in the seeds. Gas impermeability. The seed coat of certain seeds are impermeable to gases such as oxygen and carbon dioxide. Since oxygen is required for early respiratory activity in the germinating seeds, the seed fail to prolong germination. Example: xanthine. Mechanical resistance in certain wild plants such as hard and tough seed coat physically prevents the expansion of the embryo, thus they remain dormant. Example: alisma, amaranthus, and capsula, etc. Embryo induced dormancy. Dormancy due to embryo condition may be of 2 types: rudimentary or poorly developed embryo, embryo fully developed but unable to resume growth. Rudimentary and poorly developed embryo. instance, the embryos are still immature and rudimentary when the seeds are dispersed. This is seen in many species like Anemone nemorosa, Ginkgo biloba, members of Orchidaceae or Urbanaceae, etc. In such seeds, the embryo does not develop as rapidly as surrounding tissues. Embryo fully developed but unable to resume growth. In this case, the many species of example of seeds of apple, peach, iris, cherry, tulip, poplar, pines, peaks, etc. Although the embryos are completely developed in the right seed, but the seed fails to germinate even when the environmental conditions or germination are favorable. The embryo of such seeds does not germinate even if the seed coats are removed. The germination in such seeds can be induced if they are stored in moist, well aerated, and low temperature conditions. This process is called stratification or after ripening. Dormancy due to specific light requirement. The seeds of certain plant species such as Lactona sativa, Lythrum salicaria, Nicotiana tabacum, etc. have specific light requirement for germination. The light not only qualitatively but also quantitatively. The germination of certain seeds require a specific photoperiod. Example, Bignonia requires a photoperiod of 12 or more hours for seed germination. The light sensitive seeds are called photoplastic. Dormancy due to germination inhibitors. Seeds of certain plants contain compounds which inhibit the germination. Such natural germination inhibitors have been found in the pulp of the fruit, seed coat, endosperm, and embryos, or structures surrounding them. Example in tomatoes or beans or oats. A number of chemical substances such as organic acids, phenolics, tannins, alkaloids, unsaturated lactones, ammonia, cyanides, releasing substances, indoles, and gibberellins, etc. have isolated from seeds, which are germination inhibitors. Besides, other inhibitors are EBA, ammonia, phthalides, coumarin, and parascorbic acid. How to overcome the seed rot? The dormancy of seeds, even though very useful to man, is not liked by the farmers who would like the seeds to germinate soon after they have been harvested. A number of methods are therefore employed for the breaking of dormancy. The methods employed vary from species to species depending upon the cause of the dormancy. So there are main two types of overcoming of seed dormancy. One is natural overcoming of seed dormancy, and second one is artificial overcoming of seed dormancy. Natural overcoming of seed dormancy. Nature of dormancy stops when the embryo gets appropriate environment such as adaptive moisture and temperature. The seed coat of that exists in many species becomes permeable due to the rupturing of smoothing action of natural agents like microorganism, temperature, abrasion by the digestive tracts of the birds and animals that feed on these seeds. Completion of over-ripening periods. Leaching of inhibitors present in the seed coat. Inactivation or oxidation of inhibitors by heat, cold and light. Production of growth hormones which can counteract the effect of inhibitors. Attainment of maturity of embryo in case of the dormancy due to incomplete development of embryo. Then second type is artificial overcoming of seed dormancy. First type is scarification. So this method is used for breaking of dormancy of seeds caused by hard seed coats which become impermeable to water and gases, etc. The method employed in the softening or weakening of the seed coat is called scarification. When mechanical breaking of seed coat is done at one or more places, it is called mechanical scarification. Mechanical scarification is done by shaking the seeds with sand or by scratching or nicking the seed coat with knife. The treatment of seed coat with strong mineral acids or other chemicals is called chemical scarification. Chemical scarification is usually done by the dipping seed into strong acids like sulfuric acid or into organic solvents like acetone or alcohol. It can also be done